3 lectures. Today we are going to learn something about nucleic acids. Then we move to proteosynthesis. This is 2-topic lecture. Then we will have 2 lectures which will be dedicated to genetics and genetics of population. And after that, I will prepare for you the last test. You can gain the remaining points. I check the list of the points and names, so you just need to gain like 5 to 10 points. So I think it will be fine. I hope that the majority of you will be successful and I can give after this test, this last test,
Thomas,
the credits. If not, I will prepare a new test for you in January. So let's start. Let's start with
Miss,
yes.
is the quiz like next week, you mean, yeah? Or if the week
Every
after?
Friday.
Every Friday.
Every
So
Friday.
there's one today, yeah?
Today we will have, okay, this is the 2nd of December. We will have nucleic acids and proteosynthesis. Next week, 8th of December, we will have the first part of genetics. And 16th of December, we will have the second part of genetics. And after these 3 weeks lectures, I will prepare for you the test. Now it's clear?
Yes, thank you.
Perfect.
So is it better with this microphone?
Much
Yes.
better.
Much better. Okay, that's great. So maybe I will repeat it because you didn't understand me. So today we will we will go through the nucleic acid and proteosynthesis. So 2 topics today. Next week, we will have genetics, the first part of genetics. And last lecture will be 16th of December, and we will finish the genetics. And also this day, I will send you a little test, little mini test, something for 20 minutes or 15 minutes, and you can collect from this test the last amount of points. So let's start with nucleic acids and nucleus. Today we will talk about the structure components of nucleic acid, about DNA and RNA structure. So the basic difference among or between RNA and DNA is in their structure. RNA is the single strand structure and DNA is double strand structure. This is the first point difference. Secondly, we can say, please, could you switch the microphones because I listen someone. Okay. Everyone. Okay, now thank you. So the second difference is in the basic compounds from which DNA or RNA are produced. For DNA, we have cytosine, guanine, adenine, and thymine. These 4 nucleotides or its bases. And for RNA, it's cytosine, guanine, adenine, and uracil. So 3 are the same. So CGA is the same one. And for RNA, we have uracil U. And for DNA, we have T-thymine. We call these bases or nitrogenous bases, or it's aromatic compounds which are flat. They contain 1 or 2 aromatic rings, and depending on their basic structure. So we have pyrimidine and pyridine structure. So pyrimidine, longer word, refers to cytosine, and uracil and thymine, and pyridine, short name but bigger molecule, 2 circles, guanine and adenine, like the bigger bases. I don't know why it work.
Teacher. Hello.
Hello.
Something wrong? Could you listen to me, or could
No,
you see the
Miss Anna,
presentation?
I would like to let you know that we're me, Jacob, Zani, Sunny, and Min were in like from one device.
Yeah, we are recording the video. They can. I just would like to ask you if you can see the lecture, the video,
Yeah,
or
we can.
okay, no problem with my voice, no problem with the screen. Everything is visible and hearable. Okay,
Yeah.
so what's the problem? It cannot be extended, but it doesn't matter. So let's move a little bit forward. Okay, I would like to go back a little bit. So please do not interrupt me because I'm losing the idea and I will forget to tell you all what I have in my head and which I would like to tell you to. So double strand, one strand. There is also a difference in the stability. DNA, because it's much more stable than RNA, that's the third difference. The stability is bigger because it's a double strand. For example, if you provide some biochemical archaeology or anthropology or something like that, you can even find in 1,000 years old bones, usually in bone marrow, the DNA which you can amplify and analyze and read. Because DNA as a double strand, it is extremely stable. On the other hand, the single strand RNA, it's not so much stable. It's very unstable molecule. But to be honest, I have to admit that RNA, it's not really 100% a single strand because of the base pairing, there are some particular or some pieces of RNA structure which have the double strand structure simply because they are compared within the one strand. Very important is the rule based on the bases are paired. Among or between the bases are created the hydrogen bonds. Always goes A with T and C with G. For example, I have a structure here. No. Yes, here. So here you have a thymine and adenine. Between these two bases, they are two hydrogen bonds. In contrast to guanine and cytosine, where are three bases, three hydrogen bonds. This is the basic structure. And this difference arises or was firstly described by Watson and Crick. Therefore, we call this rule the Watson-Crick rule of base pairing. It's a long story of nucleic acid because firstly, the structure or the the matter which exists in nucleus was isolated in 1968 by Meissner and Hoppe-Seyler. Because they extracted this matter from nucleus, they call it nuclei. However, they didn't discover the structure. It was just a matter which was in the nucleus, and therefore they extracted. Almost 100 years later, some scientists proposed that the DNA has a double helix structure, and they 9 years later they receive a Nobel Prize. However, who discovered the basic structure of DNA, so the base pairing? It wasn't Watson, neither Crick. They just had the idea, but who really saw or made the picture of DNA was Rosalind Franklin. This scientist died as a very young to cancer. She worked with X-ray imaging of protein and nucleic acid structures, and she made a photograph of DNA. And based on the photograph, her boss, Maurice Wilkins, and his two another colleagues, Crick and Watson, suggested the structure of DNA, and they received the Nobel Prize. So firstly, the DNA structure was proposed in 1953, and 9 years later, they received the Nobel Prize. But in 1962, Rosalind Franklin was already dead. Therefore, she didn't receive the Nobel Prize, because Nobel Prize is can be you can receive Nobel Prize only when you are alive. It's not possible to give it to dead people. The science which focus on the structure, research, and functions of DNA as well as related RNAs is called molecular biology. So in these years, in the last century, the new field was proposed or started. So molecular biology is pretty young. Okay. So what are the basic structural components of DNA and as well as RNA? We call them nucleotides. You can imagine these nucleotides like a building blocks, like some bricks. These bricks, these building blocks consisted from 3 different chemical compounds. The first one, very important one for the creation of the hydrogen bonds between 2 strands, is base. We have 2 types of bases, purine and pyrimidine bases. There is also a sugar, 2 types, D-ribosis, which is present in RNA, and deoxyribosis. This one is present in DNA. And additionally is also the phosphate group. So these 3 compounds are very important to build a compatible building block, which is the basic brick for the building of DNA structure. So let's see the structures. So purines, so short name, but 2 flat circles. They are flat because these molecules are aromatic. We have some rules, which is called the Huckel rules of aromaticity, which refers or determine some rules to justify if the chemical compound is aromatic or not. Aromatic, this is the historical name, because usually these aromatic compounds, they also had a smell, so some aroma. So therefore they are called aromatic. But for the chemistry, it's much more important that they have a flat structure. So it means that they are like coins, really flat structure. No, the shape is not really it's not really 3D-dimensional practically, because it's really flat, like a coin, like this one, like this flat type. However, another rule which also arises from the Huckel rules of aromaticity is that there are not only double bonds and not only single bonds among these circles. Could you see the arrow here? So here is written that there is a single bond. This one is double bond, but this is just the formal description. In the real case, there is something in between. This is a half, 1.5, 1.5, 1.5, 1.5. Moreover, also the electrons are not really sitting between these 2 carbons, for example, or these 2, or this carbon and nitrogen, but they are flying upper the ring and on the bottom ring. So the bonding electrons are in the upper part of the ring, and these anti-bonding electrons, they are underneath of the ring. So actually it means that you have, okay, I don't have 3. So this is the molecule. This is adenine. Here are electrons flying up to the ring, and here electrons flying underneath of the ring. And we are calling these electrons delocalized electrons. Also, we can call them p electrons. P, this is the Greek name or Greek letter for p is a number. However, this is also the name for the delocalized electron. And the bonds which can be created by these delocalized electrons are called p bonds. We will need this information a little bit later. I will explain you how the structure of DNA is provided. So adenine and guanine, these two belongs to purines, so two rings or double ring structure. Pyrimidines, there are three: cytosine, thymine, and uracil in RNA. They have only one ring. They are also flat. Also, the electrons are delocalized. And just remember the Watson-Crick rule for pairing of bases. So cytosine goes with guanine and adenine with thymine or uracil. So let's see what are the difference. Sometimes in the literature you may find some words which are quite similar. So nucleoside and nucleotide. So what is the difference? So nucleotide, this is the complete brick. Nucleotide contains phosphate, sugar, and base. And nucleoside contains only base and sugar without the phosphate group. Here you can see how the structure is provided or the bonding. So here is the base, purine and pyrimidine. Here is the structure belonging to the sugar, so deoxyriboses or riboses. And here is the orthophosphoric acid residue. Here you can see that this sugar, it's a monosaccharide. Yeah. So there is one carbon, first carbon, second, third, fourth, and carbon number 5 is not in the circle, but it's really dancing outside the ring. That's very important because this carbon number 5 creates a bond with the residue of orthophosphoric acid. However, in the strand of RNA or DNA, in both of them, there is also another bond with the phosphate, and the bond occurs here in carbon number 3. However, the bond between this carbon number 3 and the orthophosphoric acid is not the original one belonging to the original building block. However, this carbon number 3 is stealing or sharing the orthophosphoric acid from the neighboring nucleotide. That's very important information because we have a strand of DNA, for example, and this strand can finish by carbon or phosphate belonging to carbon number 5 or to the carbon number 3. Based on this difference, we can distinguish the leading chain and the retarding chain. So, we have two types of chain: the parallel chain starting with carbon number 5 related with the orthophosphoric acid, and the antiparallel chain, which has the opposite constitution, and it finishes here with the phosphate belonging to the carbon number 3. So maybe you will be more familiar with this basic structure if I give you this table. Probably you know ATP, so it means that adenine as a base, there is a sugar, and P, it's a phosphate. The T means that there are 3 orthophosphoric acids or 3 phosphates. So AMP, this one, is exactly this structure. Yes, If we have ADP, there is one macroergic bond and one orthophosphoric acid more. If there is ATP, there are 3 orthophosphoric acids and 2 macroergic bonds. However, we don't have only AMP or ADP or ATP, so all the nucleotides referring from adenosine. But we have also the nucleotides, which refers to guanine, cytidine, thymidine, and uridine. We call them guanosine, cytidine, thymidine, and uridine. And also all these structures exist in the cell and they participate in the structure of DNA and RNA as well. And also we can find them in some biochemical cycles like glycolysis, Krebs cycle, etc. So let's move to structure of DNA. Please remember that this double helical structure, which is created from 2 complementary antiparallel chains, is paired based on the Woodson-Crick rule of basis complementarity. So adenine with thymine is 2 hydrogen bonds, and cytosine with guanine with 3 hydrogen bonds. And just maybe you can again use a piece of paper and a pencil and try to imagine how the structure is provided. So we have the basis, for example, adenine or cytosine, it doesn't matter. This structure is completely flat. There is p electrons and p electrons underneath. And this is another structure, like another basis. For example, this is adenine, this is guanine. And they are standing one by one in one chain of DNA. Yeah? They are slightly shifted based on the perpendicular axis of DNA, slightly shifted. There is a little overlap. If we look at that like this, you can see that they are slightly shifted. Therefore, the interaction between the p electrons can be occurred, and also the interaction exists and makes this molecule much more stable. This is the first way how the strand of DNA is stabilized. So it's the stocking interactions which are occurred via the p interactions among the electrons, which are shared with 2 bases goes by one. Also, there is another ring which belongs to riboses or deoxyriboses. It's also a circle, more or less. It's not flat out, but also it allows to create some interactions among the deoxyriboses, which are going again one by one. This is another type of structure or bond, not structure, bond. And also there is a phosphate or orthophosphoric acid, and each phosphate is shared by 2 nucleotides, one own and one from the neighbor. So therefore, there is a quite covalently or the backbone, which is based on the covalent bond among the phosphates and their sharing with the riboses. My question is, if you understand, to me, just to check that you understood, you know that the orthophosphoric acid carries, it's a residue of orthophosphoric acid. Normally, the summation is H3. Okay, maybe I will write it. I'm not sure if you can see it. Could you see the blackboard?
No.
No, okay, it doesn't matter. Okay, I will just talk. Okay, orthophosphoric acid it's normally neutral. If the acid loses two hydrogens, which are positively charged, what remains? Which charge remain to the residue of orthophosphoric acid?
Can you repeat the question, please?
Sure. We have an orthophosphoric acid. Normally, this molecule is neutral. However, if the molecule loses two hydrogen, which each of them carries a positive charge, which charge carries the residue of orthophosphoric acid?
Positive?
No, because it loses two positive charges. It should be
Negative?
because negative. Yeah, because at the beginning it was neutral. If I am losing two positive charge, there is only one option that it will be that there will be charged twice, but with a negative charge. So, but orthophosphoric acid creates the backbone of DNA. Now you know that the charge which is carried by the residue of orthophosphoric acid is negative. Could you tell me which charge will be on the surface of DNA? Because on the surface there is orthophosphoric acid. The charge of DNA will be negative. Okay, let's move a little bit. Here is the structure. We have two strands. They are paired based on the Watson-Crick rule. Here, this purple, or which color, you can see it. This is the orthophosphoric acid. It's like if I have some structure like this, and if I rotate it perpendicularly like this, I will receive more or less symmetrical structure. More or less, because there are two grooves, the major one and the minor one. It repeats, this motif repeats regularly, big groove or major groove and minor groove, major and minor. This structure, the minor groove, is very important. Usually it's the active site where some antibiotics can be bounded and they can block DNA, not only DNA of our cells, but especially the DNA of bacteria. This is one of the targeting of antibiotics to DNA of some bacteria. So just remember major groove and minor groove. Please always remember that the surface of DNA is negatively charged, and the reason is that on the surface of DNA they are fencing the orthophosphoric acids, which carries two negative charge each of them. So therefore, also what I can recognize, we have the parallel and antiparallel chains. So two chains are combined. The reason of the polarity of DNA arises from the fact that one end of the DNA strand is started with phosphate bounded to the carbon number 5, and it's finished with the carbon the attachment of phosphate to carbon number 3, and the second strand has the opposite consideration. So therefore, one of them is parallel, the second one is antiparallel. There is a difference between two processes which are occurred in DNA structure. It's replication and also transcription. Replication is always provided in the directions from the phosphate number 3 to the phosphate number 5, and transcription in the opposite order or opposite direction. So here is the structure of DNA. This is a little zoom to this double helical structure. Here you can see one strand and here is the compatible strand. So they are paired based on the Watson-Crick rule of base pairing, like this. So this is the nucleotide. Here is the base, adenine, guanine, cytosine, thymine, whatever, uracil. Here is sugar, and here is the phosphate. Okay. And we have one sequence, one chain, and this is the second one. And here is the pairing based on the compatibility and creation of hydrogen bonds. So we have two types of DNA. The much more simple and probably evolutionarily older is prokaryotic DNA. Prokaryotic DNA, it's typical for prokaryota. This is usually the single cell organisms. It has a cyclic structure like a ring or Ouroboros, no end, no beginning. However, it's not really like a circle like this, but usually it's a little bit folded mostly. Actually, we also have this prokaryotic DNA. And do you know where we have this prokaryotic DNA? In which part of our cell or cells?
The mitochondria.
Very good. So mitochondria are semi-autonomous organelles which were actually phagocytated, or we expect that it was like this. Practically the eukaryotic cell eaten the prokaryotic cell, but not digested. And mitochondria stay in cells like factories for electricity. So we are totally dependent on mitochondria. Without mitochondria, no of our cells can survive, because in mitochondria is situated, for example, Krebs cycle, beta oxidation of fatty acids, and many other processes. Quite dangerous processes, because these processes are based on oxidation. Also, the phosphorylation and creation of ATP, this is situated in the mitochondrial matrix. Also, the genes which are encoded in the prokaryotic or mitochondrial DNA, is a special name for DNA. It's a circular DNA, prokaryotic DNA, but we call it mitochondria DNA. The abbreviation is mtDNA. So the enzymes which are important for the digestion, for cellular breathing, Krebs cycle, and etc., they are encoded in mitochondrial DNA. Also, mitochondrial DNA is inherited only from mothers. It's because that during the fertilization, we have an egg, and spermia is just starting to reach an egg and get inside. They are a door, and the structure of spermia is like there is a hat. There is inside hat there is DNA chromosomes. There is a neck which is full of mitochondria, and there is flagellum which is necessary for the movement. In the neck, the energy is created for the movement. However, when the spermia reaches the door of egg, it's scissored out the neck part. Therefore, only the DNA material which is in the hat and belongs to the chromosome enter to the egg and participates on the creation of zygote. So it means that all the mitochondrial DNA which has children belongs or is coming from the mother. Based on this approach, we can build the mother trees. I will tell you more information during next two lectures, which are dedicated to genetics. Also, there is another difference between the eukaryotic and prokaryotic cell. This prokaryotic DNA is placed in the cell without any envelope, so there is not special structure for the placement of prokaryotic DNA, no nucleus, no nucleolus. And also this molecule is placed there as a free, because it doesn't create some complexes or interactions with some peptides, in contrast to eukaryotic DNA. So let's move to eukaryotic DNA. Eukaryotic DNA creates a complex with basic proteins, which we call histones. The complex, it's 8 balls. These 8 balls create a cube, and the cube serves for the coiling of DNA. Why it is necessary? Try to imagine a cable, for example, cable from your laptop, trafo, or a cable from your mobile phone or whatever. If you put this cable together with another cables you have into one case, to one special tiny space, what happens? If you would like to use one trafo, you have to open it and remove practically the whole ball of your cables. The same problem would occur in your nucleus if you put there such a long molecules of DNA into pretty more small tiny space. Therefore, it means that you have to coil DNA and put them or condensate it to a smaller and tiny, more defined particles, shapes, which we call chromosomes. This condensation of DNA, this is not provided randomly. There is a quite strictly way how to coil or condensate DNA to these histones. It looks like that. So at the beginning, we have DNA double strand. It's coiled on the histone complex, on the cube created from 8 balls, and it is coiled and coiled and coiled again like this. Maybe this picture is slightly better. You can imagine how the coiling is provided. Maybe I can show you a little video just to know how it works. Do you know coiling gizmo? So this is the first step of coiling. So you can create, this is the single coiled structure, the same like we are coil DNA to histones. This is the second level of coiling, and this is the third level of coiling. So the principle is like this. This is the first level coiling. You can imagine it like quite pretty easily. And okay, this is the second level of coiling. Okay. But it's not enough for DNA. It has to be coiled a little bit more like this. Okay, is it clear how the DNA is practically coiled or condensated to more defined structure, which is called chromosome?
Yes.
Thank you for some feedback. Okay, this is a little advertisement or invitation because majority of you are foreigners. Not okay. There are 2 students, maybe 3 from Slovak, 2 from Czech. But have you ever been in Žďár nad Sázavou? It's here in Vysočina. This is okay, we have mountains here and here, but also this is the mountains which are in the middle of the Czech Republic. This landscape is pretty special because it was quite empty space for a long time, and people who started to live there, there were many forests, and people were a little bit afraid, or some ghosts and elves in the trees and so on. Therefore, in the 17th and 18th century, there were built many architecture which is related to Christian architecture. Very special one is from this Jan Matěj Blažej Eichel Santini. This guy was very famous architect in the Czech Republic. He built up or designed many Christian architecture, and this one is pretty special because he uses pretty strange symmetry. If we look from the top from a drone to the architecture of this is a cemetery and a church in the middle. If you look at the structure from the architecture plan from the from drone from the upper view, we will see that this structure is pretty similar to circular DNA. This is pretty scary. Definitely, Santini didn't know anything about DNA. He didn't know the structure. He didn't know that we have cells, and that in the cells that we have DNA. That was just his intuition, and he built this amazing architecture. Actually, this place is very special because I was there several times, and I never felt that it's a scary place, because this is the graves of monks. This is cemetery, and in every May we celebrate the Saint John. It was a patron of the Czech Republic as San Wenceslas, so St. John is also our patron, and there is a big celebration. I think it's 16th of May. So if you have time, I can strongly recommend you to visit it, but do not visit it during the night, because this is a very scary place. So that was a little invitation. And now we can move to functions of DNA. So DNA and RNA, they have different functions. The function of DNA is mostly reproduction and transfer of genetic information. So two basic functions. For RNA, it's not so easy to describe it because we have several types of DNA. It's messenger RNA, transferred RNA, ribosomal RNA, at least these three basic ones. We have also microRNA and also something medium-sized messenger RNAs. And now we know from some, it's not recent research, maybe 20 years ago, and the Nobel Prize was given 2010, I think. So the Nobel Prize was received by a person which name is Thomas Čech. So probably you know that what is his origin, and he received this Nobel Prize for the discovery of some special function of RNA. And he discovered that RNA has not only this genetic or encoding function which participates on the production of proteins, proteosynthesis, which we are going to speak a little bit later. And he discovered that RNA also behaves like an enzyme. So it has a regulation function. It's enzymatic, so therefore the RNA probably was the first nucleic acid which exists, and also it helped, or it had the same functions like DNA, because the RNA can be replicated as well. Moreover, RNA has the regulation function like enzymes. So this is very important, and it's pretty new. So steps which are included in DNA replication. First of all, we are going to speak. Replication. And so there are two processes which are important for our cell. It's DNA replication, which is related to mitosis or meiosis. and also transcription and translation, which are two processes which belongs to proteosynthesis. And these two processes are necessary for the synthesis of proteins. Every cell needs proteins, and proteins are provided by cooperation among DNA and RNA. There are some enzymes which are necessary, but I will skip it a little bit and I will move to structure of RNA. I don't know why it is switched. So what is the basic difference in the structure of RNA in contrast to DNA? For the first view, there is a difference. We don't have thymine, we have uracil instead of. Also, it's not double strength, but just a single strand structure. But it's not 100% true, because among the single strand structure, we can observe something like stem and hairpins. So stem and hairpins, this is the structures which has the character of the double strand structure. However, this double strand structure occurs among this single strand molecule. We have three basic types of RNA. This is tRNA. It's a pretty small RNA. It looks like this. There are these three hairpins or lobes. It's not hairpin, lobes, and also these four necks or stems. This side, in one end, this one with the red color, it contains three bases. It's a sequence of three bases, which is called triplet. Exactly, this triplet is called anticodon. This anticodon structure, triplet structure, is compatible with codon, another triplet structure, which is placed in messenger RNA. By a complementarity between codon and anticodon, the placing of amino acids to the first amino acid structure or the amino acid structure or a single strand of newly created protein, which is placed here, the creation of the chain is provided. So there are two sides. This one, which creates the compatibility and occurs the placement or correct placement to the newly created oligopeptide chain, compatibility between codon and anticodon. And also here is the active place where the amino acid is bounded. The second type of RNA is ribosomal RNA. It's much more bigger, and it creates the basic part of so-called ribosome. This ribosomal structure is very important. It's created from proteins as well as from RNA, and especially these active sites, these hairpins and lobes, it is expected that they behave like an enzymes. It means that the ribosomal structure catalyzes the proteosynthesis. And the main function of RNA wasn't clear so far, so many scientists should to discover what happened exactly there. Messenger RNA, this is the template. Where one gene is rewritten during the process which we call transcription. So transcription occurs in nucleus, and then messenger RNA get into cytoplasm, became in close contact to ribosome, to the small ribosomal unit, and based on the compatibility between tRNA carrying anticodon and codon, combination of three nucleotides, the placement of every amino acid is provided one by one. Now we are getting to the functions of RNA. So the function of messenger RNA is that it codes the position of each amino acid based on the codon and anticodon interaction. tRNA specifically select the compatible select the specific amino acid based on the character of anticodon. And ribosome RNA, it catalyzes proteosynthesis, it bounds messenger RNA and tRNA together and somehow provide the interactions between these two molecules. So this is the first part of the lecture. Now we can move to proteosynthesis. Do you have some questions to the topic? Okay, if not, I'm happy that everything is clear. Why we cannot? Okay. So proteosynthesis. This is the process which is important for synthesis of protein, for the biosynthesis of protein. So proteins are us. We are made of proteins. So how we look like, the proteins are responsible. And what predetermines how our proteins and we look like and we are, it's our genes. So in our DNA, we have 23 pairs of chromosomes. Every chromosome contains hundreds of different genes which encode proteins. So this DNA, which codes some proteins, is called coded DNA. Actually, this coded DNA amounts 5% from the whole amount of DNA. So the last 95%, so the majority of our DNA, we actually don't know the purpose. So it is the wastelands of DNA. This is the non-coded parts of DNA. And the researchers really, they are really curious to find the main reason why we have such a long parts of DNA and we don't know the purpose. Probably there is a purpose, but we don't know yet. We didn't discover it. So what we are going to do? First of all, we will introduce some basic parts of the proteosynthetic apparatus. We will talk about the mechanism of proteosynthesis, and we will describe briefly co-translation and post-translation adjusting of protein. So first of all, what is needed? Just try to imagine that you would like to cook I don't know, a dinner or summer or a Sunday lunch. What you will need? You will need a matrix. First of all, the matrix is placed in DNA. It's a piece of DNA. This matrix, but because DNA is never going outside nucleus. It's always placed. Try to imagine the nucleus like a library. It's a library which is closed. You can enter there and you can just make a copy of one of the books and leave the library with the copy of the book. You cannot take the book from the library. Books are stored in the library. This is very secret books, very treasure. It's a pressure. It's a pressure, our pressure. So DNA, it's covered, protected in our nucleus. So the process which we are making the copy of one gene, it's transcription. So the template from which the piece of DNA is rewritten is messenger RNA. So the structure of messenger RNA refers to the structure of the protein. This is 4.5 billion years old alphabet. Try to imagine that we have a language of proteins and we have a language of nucleic acids. And we need some translation between these two. And this translation is provided in cytosol, in special structures, which we called ribosomes. And we need something which we call the translation factors to help to translate the language of nucleic acid to the language of proteins. Also, to the synthesis to proteins, we need the basic substrate, and the basic substrate is activated amino acid. Activated why? So why I'm talking about not only about amino acids, but I'm talking also about activated amino acids. To create a bond, this is usually a very demanding process. I mean, the energy demanding process. Therefore, these amino acids should be activated to create the peptide bond, because the peptide bond is very strong bond, and therefore I have to add a lot of energy to promote the bond creating. So how it looks like. This is okay. This is nucleus where the transcription occurs. Close to the nucleus, we have something which is called RIV, the endoplasmatic reticulum. On the surface, on the endoplasmatic reticulum, we have ribosomes. Ribosomes are the units which serve for the biosynthesis of proteins. However, it's not enough to synthesize the polypeptide chain. We have to provide the secondary, tertiary, and quaternary structure, which is provided in endoplasmatic reticulum. And also we have to provide some packaging of newly created proteins, and they'll deliver it to the target place. So how it works together. Okay. So let's focus on ribosome. So what happened? So at the beginning, we synthesize the compatible chain of messenger RNA from a gene which is placed in our nucleus, in our DNA, because DNA never is never be replaced from nucleus. We can call ribosomes as a factories from proteins. If we dry cell, they provide something like 1/3 or 1/4 to 1/3 of whole dry cell matter. So it's really frequent structure within the cell, whole cell structures. It has two structural units, the big structural units and the small structural units. Approximately 2/3 of ribosomes is created by ribosomal RNA, and the last third is created by proteins. The interconnections of these two structure compounds is provided by a crossing of magnesium cation. So how the process is occurred? So this is the small ribosomal unit. This is messenger RNA. This messenger RNA is captured on small ribosomal subunit, and also there is a tRNA. Here, this part occurs the. Potřebujete něco? Já vydrž chvilku, jo? Já ještě půl hodiny učím. Super, dobrý. Ale se omlouvám. Dobře. Okay, we have a tRNA structure, which is that one. And there are two main functional parts of tRNA. This is anticodon. Anticodon is represented by the triplet. Triplet is the sequence of three letters, three nucleotides. And here is also the place where the concrete selected amino acid is captured. So we have something like 20 or 21 amino acids. You know it from biochemistry. So we have something like tyrosine, leucine, valine, histidine, methionine, whatever, 20, 21. And these amino acids are selected by their tRNA, and based on the compatibility of this anticodon with the codon structure in the ribosome, the amino acid is placed to the creating polypeptide chain. On the big subunit of the ribosome, there are two places. This is P place, where the newly created polypeptide chain is created. And here is A place or A site, where the newly coming tRNA with new amino acid is just starting to be attached, and the combination or complementation between anticodon and codon is provided. So this is just the rough picture of the whole process. Trust me that it is much more complicated because the corrections has to be provided. The matchmaking system is quite sophisticated. The translation factors, which knows the language of amino acid as well as the language of oligonucleotide or the nucleic acid, has to be presented and included into the process. But just to understand what happened, the principle would happen. This is, I think, enough. So this is just the by word transcription, what I show you on the picture. So firstly, maybe I can skip it because I already mentioned it. And I would like to show you how the activation of amino acid is provided. I mentioned at the beginning that amino acids, that they are not really willing to provide some proteins. They has to be a little bit pushed and motivated to create the oligoprotein chain or oligopeptide chain. Therefore, we had to add a lot of energy to destabilize the amino acid and to invoke or provoke the creation of the bond. So in the first step of translation, we have to enzymatically convert amino acid to amino acyl tRNA. So it means that I have amino acid and I have tRNA, and I have to enzymatically attach amino acid and with their tRNA. For this enzymatic reaction, I need an enzyme which is called amino acyl tRNA ligase. So ligase, something put together. Probably Isaac already mentioned to you enzymes, some lecture for enzymes. So you know that ligases, this is one group of enzymes which are frequently used in biochemistry. Also, what happened there, the reactivity of amino acid is increased. So the amino acid is provoked to create a peptide bond, to create a bond with another amino acid. Also, the selection of appropriate amino acid, which has to be placed in exact place depending on the recipe which is written in messenger RNA. It's provided by the selection of tRNA. So two separated processes, activation and selection, because the position of amino acid in the polypeptide chain, this is very important. This is the question of primal structure of protein. And the primal structure of proteins also influence the secondary, tertiary, and quaternary structure. And therefore, in the end, it may affect or somehow influence the resulting structure and function and working ability of protein. So it can happen that if some mistake during this process is provided, we can synthesize an enzyme or a protein with the wrong structure, unfunctional enzyme, and therefore some problem, some sickness, diseases, illness can start. We also have among the population several variants of genes, so therefore we have several variants of proteins. We have we have two copies of genes, one from mother, one from father. Therefore, we have alleles. Alleles is the variant of a gene. every one of us has a two alleles, two variants of gene, one from mother, one from father. It can happen that you have the same alleles from father as well as from mother. For example, I will explain it to you on the problem with red blood groups. Probably you know that we have among the population, we have blood group 0, A, B, and AB. Every blood group is created as a combination of two genes, of two alleles. If you have blood group 0, you should have a genes or an allele from father as well as mother, which refers to 0, because this allele is always recessive. So we can call you a homozygote. If you have B, there are two options. You may have the same alleles from your father and mother as well, and you are homozygote as well. Allele B as well as A, they are dominant all the time. There's a second option. If you have B or A, you may be also a heterozygote. It means that you have one allele from father, like B, for example, or A is the same, and 0 from your mother, for example. If you combine it together, because 0 is always recessive and A or B, they are always dominant, the result will be that you have a blood group A or B, depending on the dominant allele. If you receive one allele B from father, one allele A from mother, for example, because both of them are dominant with the same level, your blood group will be AB. So this is just briefly the background of blood groups. So if I conclude it, among the world population of humans, we have genes for 0, A, and B. So just 3 alleles, 3 types of gene among the whole world human population. Another story is about, for example, insulin. Insulin is also a protein, and it is encoded among the population. So we have some variants among the population. And also there are some genetic diseases which arises from the wrong combination or wrong sequence or some mistake in the sequence in the genes, and therefore this is the diseases which cannot be easily solved. Yes, we can use something like genetic scissors, and in the stage of embryo, something has to be repaired. But the diagnostic is very complicated, and there are also some ethical aspects and so on. But in the principle, we are able to repair the genes, and therefore to heal some genetic diseases from this way. But not only the genetic disease occur, but some mistakes during the transcription because of the pollution, radiation, and so on may occur, and therefore our body is producing the wrong copies of proteins, and therefore if they are wrong, they cannot work properly. So I will give you more information next time, because this is the topic which is related with genetics. How it works. So first of all, I have amino acid. I need some energy, ATP. I need the enzyme. And in the end of this catalytic reaction, I have amino acid, which is bounded to via phosphate ribose or became activated. So this complex is called complex amino acyl adenosine monophosphate enzyme. Later on, this complex became matched with tRNA. So in the end, I will have a tRNA together with phosphate ribose adenine and amino acid. We call it amino acyl tRNA or activated amino acid. This complex is now willing to enter to the biosynthetic reaction and create the newly created polypeptide chain. So I think this is not important. Yeah, but there is a difference among eukaryotic and prokaryotic DNA and RNA. Yeah, no, but I would like to mention something else. I don't remember if I describe you the difference between eukaryotic and prokaryotic DNA, because did I talk about telomeres? I don't remember. Not in this lecture, but maybe in the past. I can talk about it later. I think so. So, okay, this is very crucial. So try to imagine that I have messenger RNA. The messenger RNA is a template for the production of one protein. There is a sequence of nucleotides, triplets, and so on. This messenger RNA is captured in ribosome. Actually, normally not in one ribosome. There are many of ribosomes, thousands or at least hundreds in one movement. But try to imagine just one ribosome, one messenger RNA to be sure that you understand the process. Based on the recipe written on messenger RNA and selection of amino acids with DNA, the newly created chain is done. Try to imagine that I have a long sequence of amino acid, which is called oligonucleotide or oligopeptide, or the primer structure of proteins. This primer structure of proteins is not really the functional protein. It has to be provided many reactions to finalize the structure of protein. And for this finalization of the structure of proteins, we have two structures among the cell. We have endoplasmatic reticulum, roof endoplasmatic reticulum. So the newly created polypeptide chain because of endoplasmic reticulum, and inside releases. So from biochemistry, we have two amino acid which carries the sulfate. Could you name them? Did already Isaac spoke or taught you amino acids and proteins? Or not? Okay, it's methionine and cysteine, and the bond
Your
which is created
microphone
is
keeps
disulfide
breaking up.
bond. It's very important to remember this.
Where did she go?
No idea.
Is she back?
Yeah, I'm back.
Yeah.
Sorry, I don't know what happened. So presentation.
Your microphone kept cutting out, and then you disappeared.
Okay.
You asked a question, and then you went away.
Okay. And now you can hear me at least.
Yes.
Okay, perfectly. So I will share the presentation again. So, okay. happened? Friday? Okay. Now you can see endoplasmatic reticulum, yeah? So I spoke about cysteine and methionine, two amino acids which contain sulfur. And in endoplasmatic reticulum, they are provided the bonds between these two sulfates, and it creates disulfidic bonds. It's a type of a covalent bond, and usually within the structure of the protein, these disulfidic bonds match together and they create the active sites of the protein. So usually it's the active site of the newly created enzyme, protein, whatever. Actually, also some metals can be attached, sugars and lipids to provide a more complicated structure. It can be also done, and it is also provided in endoplasmatic reticulum. But just within the structure of the protein, the disulfidic bond, this is very important, and usually it is the active place of the enzyme or protein. Also, the chain, which is newly synthesized and it's pretty long, it's slightly cut or modified, the side chains. It's not necessary that this just says single strand structures, but there are some side chains. And these side chains, they made some bridges within the structure. So it became not just the strand structure, but no more branched, and in the end, more global or some spaced, important or interesting structure. All these reactions are done in endoplasmatic reticulum. What's also done is the attachment of non-peptides. So I named them sugars, lipids, and some metals or phosphates also. Usually these 3 or 4 groups of non-peptide chemicals is attached. So these all chemical reactions, this modification is provided in endoplasmatic reticulum. So in ribosome, I receive the primal structure of the protein. In endoplasmatic reticulum, secondary, tertiary, and quaternary structure is provided usually. Sometimes the quaternary structure can be also provided on the target place, usually in the plasma membrane. But to transport the protein, newly created proteins, I need some like Česká pošta or some something to deliver. So I have to make a package of the protein. So there is something which separates peptides and put together the same type of proteins to a vesicles, so packages. So the packaging, this is the aim of Golgi. And in these vesicles, in these packages, the proteins are transported to the target place. So the target place can be in the range of the cell. So usually we synthesize proteins to become a part of plasma membrane as a channel, as a transporter, carrier, but it can be also placed in some different part of the tissue or in the body. As an example, the Langerhans islet, they produce insulin, but insulin, his target is not in the insulin islet. Mostly his target is in another parts of the body. So it is transferred via the bloodstream to every single cell. So 2 types. It remains in the cell or within the cell, or it is transferred via the bloodstream to different parts of the body. So, little conclusion. We have just something like 8 minutes to finish it. So, at the beginning, we have DNA. This is the DNA. This is the double strand structure, which is always placed in the nucleus. Never be released from nucleus, always remain in the nucleus. So DNA is lazy, so it stays in nucleus. It's not moving, it's not migrating. But if our body needs to synthesize a protein, the body needs a recipe. So the recipes are written in the book. The book is our genes. This is our bibliothèque, our library. But the books are not leaving library at all. Therefore, I have to visit the library and rewrite the recipe to a paper, to a special sheet of paper. And only in this form, I can transfer the recipe from the library. So DNA in the nucleus serves as a library. Messenger RNA serves as a template for the transfer of the information from DNA to messenger RNA. The process of the rewriting of the information from DNA to messenger RNA is called transcription. Transcription. It's in Czech, but it's the same. So transcription. This transcription is provided on the principle of the pairing of bases. Here you can see A goes to U, C to G, C to G, A to U, U, 3A, C, G, C, G, GC, AU, GC, and TA. So the same compatibility. As I mentioned, T in DNA, U in RNA goes to A or refers to A or is matched with A only. G always is matched with C. Every triplet, every sequence of nucleotides refers to the to one of the 20 amino acids, coding amino acids. There is, I don't have the table here. In biochemistry, probably Isaac shown you a table where it's visible that we have a code, like a codon in messenger RNA, like UGG. And in the table, you can find that this codon, this triplet, refers to tryptophan. Based on this matchmaking, tryptophan is selected by tRNA. The anticodon on tRNA is compatible with codon, which is on messenger RNA. So AU, CG, CG again. During translation, which is the process which occurs in ribosomes, the oligopeptide chain is synthesized based, or the sequence is synthesized, the primer code is written in messenger RNA. So the protein has the exact content, exact sequence based on the information which is written in messenger RNA, but it originates in DNA. Is it clear, this process? Just raise a hand if this particular process or this, if this is clear. I mean, raise a hand like this. Okay. Tara, Marcos.
It's clear.
But okay, clear. Okay, at least something. Thank you very much. So in the end, last slide. So in nucleus, what happened? Transcription. So the transfer of information, the transcription of gene to messenger RNA. In cytosol on ribosomes, the messenger RNA serves as a template, as a precursor for as a template, as a recipe for proteins, peptides. Because of the participation of tRNA, all the amino acids are placed to the polypeptide chain. So it means something like prepeptide or precursor of a peptide is created. However, this structure is not working. This is not functional. Therefore, this precursoric peptide is transferred to endoplasmatic reticulum, where it is chemically modified. And then, which is missing here, goes to the Golgi, where the packages are created. And in these packages, the proteins are transported to the target place within the body. So that's all. I will also send you the video. This one, you can see the animations. So I'm putting to the chat, and also once the video became recorded, I will send you this video from this video lecture. Questions? If not, I wish you a nice weekend, and I am looking forward to another online lecture. So next Friday, now I think it's 9th of December, from 1 p.m. we will have the online lecture, which is dedicated to genetics. So thank you very much for your participation, and have a nice weekend. Bye-bye.
Bye bye.
Thank you.
You too.
Bye.
Thank you.
Thank
Bye-bye.
you. Bye.